There are now thought to be more than 35 million sufferers of Alzheimer's worldwide, and this is expected to increase to 115 million by the year 2050. Alzheimer's is the sixth largest cause of death, and unlike the other leading causes of death, no proven cure, prevention, or means of slowing Alzheimer's disease has been found to date. Researchers at the University of Minnesota have been developing new ways to identify and prevent Alzheimer's disease.
I'm Swati More. I'm an assistant professor at the Center for Drug Design, University of Minnesota. We got started with the Alzheimer's project based on the research we were doing on Parkinson's disease. Dr. Vince and I realized that the methods we have been using for Parkinson's disease could also be promising for the Alzheimer's disease research.
This approach to Alzheimer's disease considers two events that occur in the brain, the first being the brain's ability to process sugar. When sugars in the brain are oxidized, they form a toxic molecule known as dicarbonyls. However, the brain contains an enzyme known as glyoxylase, which clears the brain of these dicarbonyls. In the early stages of Alzheimer's, the brain loses its ability to produce this enzyme that clears the brain of the toxic molecule. The second event that occurs in the brain of Alzheimer's patients is the unraveling of a peptide known as amyloid beta, caused by the attachment of the dicarbonyls. When the amyloid beta unwinds, it forms what is called a B sheet. When multiple B sheets are formed, they will begin stacking on top of each other. They will continue to stack, forming plaques in the brain. This causes the effect of Alzheimer's disease. Past attempts at combating Alzheimer's have focused on eliminating the plaques in the brain after they have formed. This approach takes into consideration the early stages leading up to the plaque formation.
Using this approach, we've developed a candidate drug that was designed to stimulate the action of a glyoxylase enzyme to help it get rid of these toxic dicarbonyls. And when we test this candidate drug in transgenic mice, we found that the drug will actually prevent the mice from getting to the plaque stage, where no plaques are actually developed in the brain, and the mice actually retain all of their ability of memory. And we feel that this process might be useful in treating a person who is progressing toward Alzheimer's disease, instead of waiting until symptoms develop and plaques develop and then try to intervene at that point.
Early detection of Alzheimer's disease is very important. We have found that we can detect the unraveling of amyloid beta peptide that happens in Alzheimer's brain by scanning the retina of the eye. This gives us an excellent method for detection of Alzheimer's disease progression. In our early experiments in transgenic Alzheimer's mice, we could detect this Alzheimer's progression by retinal scanning. An advantage of this new retinal scanning technology is that it is non-invasive. Unlike current methods, no extraneous dyes are required, and the changes that are occurring in the brain can be seen without directly looking into the brain. This new technology has practical application in that it could be performed during a regular eye exam. These new technologies are the first steps towards early intervention in Alzheimer's disease.
This approach could hopefully lead to the development of drugs that could be used for early onset Alzheimer's.
For more information about these Alzheimer's disease technologies, contact the University of Minnesota's Office for Technology Commercialization.